Hello dear students, today we are going to learn the complete concept of heredity, which is very important for your board exams. Now starting with the Mendel's law of experiment to monohybrid cross and dihybrid cross, everything I'll explain in detail but in simple way. So before I start the chapter, let's try this question. If a tall plant is crossed with a dwarf plant, what do you think the offspring will look like? Comment your answers in the comment section below. Okay, now let's understand the definition of heredity. So what is a heredity? Heredity is the transmission of traits from parents to their offspring, and that is through genes. These traits are also known as the characteristics. Okay, so in simple words we can say that it is the passing of feature from one generation to the next, that is from parent to a child. Understood? So this is heredity. And now what is a trait? Trait is basically the specific characteristic that is present in an individual. Like for example your blood group, right? Or all are not having the same blood group. Some are having O blood group, AB. So these are the blood group, or these are the specific characteristic that is having in an individual. Then eye color, hair type, skin color. So all these are coming under the traits. And these traits are classified into two types. Now what are they? Inherited traits and acquired traits. Inherited means what? They are passed from parents. Okay, and acquired traits means they are developed during the lifetime. Like for example injury, right, or any scars that develops in your hand or your leg. So all these are acquired during your lifetime. Inherited means that is inherited, or that is passed from the parents to the offspring. Clear? Okay, now how do these traits get inherited? Traits are inherited basically through the genes that is present on the chromosomes. Now during reproduction process, one allele for each trait is passed from parent to the offspring through the gametes formation, and during gamete formation the alleles separate. Now this we can take as an example of a plant height. Right, in plants we can see tall and dwarf plants, right? So capital T and capital T is assumed as a tall plant, whereas small t is a dwarf plant, right? Now the gametes that is formed over here is capital T for the this parent plant, that is a tall plant, and for the dwarf plant it is small t, right? Now when they fuse together, what they form? Capital T and small t, right? So this is tall, right? This is what it is said here. During reproduction, one allele for each trait is passed from each parent to the offspring through the gametes. Now when the male and females fuse, one allele from the father, that is this one, okay, one allele from the father and one allele from the mother, they gets fused, and the offspring gets these two alleles, that is capital T and small t. Okay, so this determines the trait of a child, and at fertilization process the alleles combine, determining the traits of an offspring. Got it? Now let us study about the genetics. Now what is genetics? Genetics is the branch of biology that deals with the study of heredity and variation. And who is the father of genetics? Father of genetics is Gregor John Mendel. Okay, Gregor John Mendel is known as the father of genetics, and he performed the experiments on pea plant and discovered the basic laws of inheritance. Okay, and the scientific term of pea plant is Pisum sativa. Okay, this one, this is important for your exam. So if a scientific term of pea plant is asked, the answer will be Pisum sativa. Okay, now why did Mendel choose only pea plants for his experiment? Why not any other? The answer is, okay, the correct answer is that is pea plants are easy to grow. Okay, and they have a very short lifespan, that is 3 to 4 months. Okay, so they have only 3 to 4 months lifespan. They have a clear contrasting traits. It is self-pollinating. Self-pollinating means what? What is pollination? Pollination is the process of the transfer of pollen grains from anther to the stigma. Okay, self pollination means, self means what? Within the same flower. Okay, so self pollination is the transfer of pollen grains from stigma of the same flower to the anther of the same flower. Okay, then cross pollination we can do it, but we can do it in an artificial manner. Otherwise the plant have a self pollinating process. That's why Gregor John Mendel chose pea plants for his experimental purpose. Now let us see what are the contrasting triads in the pea plant. As you can see here, that on the left side you can see that the triads, dominant triad is there and the recessive triad. Okay, now when we take the shape of the seeds, the shape of the seeds are two, right? Round and wrinkled. Round is the dominant one and recessive one is the wrinkled. Then color of the seeds, color of pods, shape of pods, plant height, position of flowers and flower color. So all these are the 7 pairs of contrasting triads that is seen in a pea plant. Students, this is very important for your board exam. You should study the contrasting triads in a pea plant. What are the 7? Then let us understand the basic important terms that comes in genetics. What is a gene? Gene is a unit of heredity that controls the triad. Okay. Then chromosome. What is chromosome? Chromosome is a thread-like structure that is present in the nucleus that carry the genes. Nucleus is the brain center of the cell. Okay, so the chromosome is the thread-like structure that is present in the nucleus and that carry the genes, that is the DNA. Then what is a triad? As I said earlier, triad is the specific characteristic of an organism. Okay, now what is an allele? Allele is an alternative form of a gene that controls the same triad. Or we can say that allele is the different version of the same gene. Like for example, ice cream flavors. Okay, let us take an example of an ice cream flavor. So here we can say that the gene we can consider as ice cream. Okay, so gene is the ice cream, whereas the alleles, okay, the alleles is the chocolate or vanilla. Okay, as I will consider as these two flavors in the ice cream. So gene we can consider as the ice cream, the alleles are the chocolate or vanilla. Now both are ice cream, right? Both are ice cream, whether it is a chocolate or vanilla, like both are ice cream. So this is the same thing, but they have a different flavors. Chocolate and vanilla are different flavors of the ice cream, right? So these are different version. Like that, the gene is the triad, allele is the different version. Okay, understood? So what is an allele? Allele is an alternate form of a gene that controls the same triad. Now alleles are of two types, dominant allele and recessive allele. Now what is a dominant allele? Now what is a dominant allele? Dominant allele expresses itself if only one copy is present. Like for example, if we take an height of a plant, capital T, okay, so this is tall, right? So this is dominant one because it expresses itself. Or we can say that if it is capital T and small t, in this case also this is dominant. Why it is dominant? Because it expresses itself even only one copy is present, and it is always represented by capital letter. So always remember that if a capital letter is present, then that is dominant. Okay. Now what is recessive? Recessive means it expresses only when both alleles are recessive, and it is represented by a small letter like small t and small t. Okay, so small t, this is recessive. Why it is recessive? Because it is represented by a small letter. Okay, so it expresses only when both alleles are recessive, that is, it is hidden. Okay, so that is recessive. Now let us understand what is a genotype. Okay, one of the most common term that is used in genetics is genotype. Now what is a genotype? Genotype is the genetic constitution of an organism for a particular trait. Genetic constitution, or in simple words we can say that it is the genetic makeup. Okay, a makeup of an organism, or the allele combination of an organism for a particular trait. Again let us take an example of a plant height. Okay, so you already know that in plants there are two heights, right? A tall plant and a dwarf plant. Okay, and in tall plant it is associated with capital T, and in dwarf plant it is small t. Okay, now what are the allele combination in tall plant? This is capital T, and in dwarf plant it is small t. So this one, this capital T, small t, this is the genetic constitution of a particular trait. Okay, so this is genotype, capital T, this is genotype, and this is also a genotype. Now what is a another important term is phenotype. Now what is phenotype? Phenotype is the observable physical expression of a trait, that is we can see externally, right? What we can see externally, that is the phenotype. And here what is phenotype? Phenotype is the tall plant, right? When we see outside, the plant is tall, right? It is tall, it is dwarf, right? So this is phenotype, the one which we can see out, externally, that is phenotype. So the tall, this dwarf, this is phenotype. And what is genotype? The genotype is the one which is present inside it, right? The like capital T, small t, all these are the genotype. Am I clear to you? So this is the basic difference between a genotype and a phenotype. Genotype is the genetic constitution of an organism, while phenotype is the one which we can see externally. Okay, and in this there are other two terms, homozygous and heterozygous. Now what is homozygous? When both alleles are same, as we see here that capital T, or small t, this is homozygous. Why it is homozygous? Because both the alleles are same over here. Okay, now in heterozygous condition the alleles are different, like capital T, small t. So this is heterozygous condition. Why it is heterozygous? Because the alleles are different, right? One is capital and the other is small. So that's why it is heterozygous condition. Clear to you? So homozygous when both the alleles are same, and heterozygous when both the alleles are different. Okay. So these are some of the important terms that is associated with the genetics, and please study for the examination. Okay, now let's see the difference. What is monohybrid cross? Now what is a monohybrid cross? Monohybrid cross is a cross or a pair that involves only one pair of contrasting traits. What is contrasting traits? Traits means capital T, or small t, So this is a trait, right? So the pair that involves only one pair of contrasting traits, resulting in 3:1 ratio. Okay, like for example, if we take an example of a height of a pea plant, the tall plant, right? Tall plant, the trait is capital T, and in dwarf plant it is associated with small t, that is dominant and recessive. Now let's see how we can calculate, or how we can do the monohybrid cross. Okay, so let's solve what is a monohybrid cross. So in monohybrid cross, as I said, we have to take an example of a what height of a pea plant. So in the plant, what are the two main traits? The tall plant and the dwarf plant, right? So this is the phenotype. And the possible the parent genotypes, what are the parent genotypes here? Capital T and small t. So this is the genotype, and from this we have to find out the gametes. Now in tall plant, what is the possible gametes? It is capital T. Okay, that we will make it in round. Please remember, you have to round it here. Then small t. Okay, so these are the gametes that is formed for the tall plant and the dwarf plant. And then what we need to do, we have to cross it, right? The capital T and small t, and when we cross these two, what is the resulting what product? That is capital T and small t. We get capital T, small t, and this is the F1 generation. Okay, so these tall and dwarf, these are the parent. Okay, this is the parent gamete, and this one is the F1 generation. Okay, now next is what we need to do in the next step. We have to do self pollination, as Mendel, what he suggested. Why did he do this? Because the plant is self pollinating. If we want to find out the F2 generation, then we have to self pollinate it, right? So here we need to do the self pollination, that is, we need to cross this with this one. Okay, so capital T, small t, capital T and small t. Okay, so self pollination we have to do. So when we do the self pollination, again we have to find out the gametes. Now what are the possible gametes over here in capital T, small t? What we get it? We get capital T and small t. Okay, now here also we get capital T, small t. So these are the F1 gametes. Okay, now from here we need to do the crossing, that is, this is a Punnett square where we need to do this. Okay, so I have made already a table here of 4. Why? Because these are the 4 gametes, right? So capital T I will write here, then small t here, because this is male, this is female. Okay, then capital T and small t. Now what is the next step? We need to cross these. So capital T and capital T here, then capital T and small t here. Again we have to cross this, capital T, small t, then small t, small t. Okay, okay, so what is the resulting product here? Now capital T, what is capital T and capital T? It is tall, what we can see externally, right? And this one, capital T, small t, is the tall plant. Okay, why it is tall? Because one capital letter is there, right? Capital T is there, that's why it is tall. Then in the next one, capital T, small t, again it is tall, and then small t, both are recessive genes, right? So this is dwarf. Okay, now when we take the ratio, like phenotypic ratio, what is phenotypic ratio? The phenotype what? Phenotype is the external, right? The which we can see externally, right? Like tall plant. Now how many tall plants are there? 3, right? And how many dwarf plants are there? 1. So what is the phenotypic ratio? It is 3:1. Okay, now next let us take the genotypic ratio. So genotype means what? Genotype means the one which we can see it inside, right? Like capital T. How many capital T are there? 1, right? Now capital T, small t, we have 1 and 2. Okay, and small t, 1. So what is the ratio here? 1:2:1. So this is the genotypic ratio of the monohybrid cross. So the ratio of the monohybrid, or the phenotypic ratio of monohybrid cross, is 3:1, whereas the genotypic ratio is 1:2:1. Okay, clear? Okay, now let's see what is a dihybrid cross. Now dihybrid cross is a cross that involves 2 pairs of contrasting triads that results in 9:3:3:1 ratio. Okay, so di means what? 2. That's why it involves 2 pairs of contrasting triads, whereas monohybrid, in that only 1 pair was there. Okay, but in dihybrid cross there are 2 pairs that results in 9:3:3:1 ratio. Now let us take an example of like a pea plant. In pea plant there are like two contrasting triads we will take, seed shape and seed color, right? In triad 1 we can take right, round and wrinkled, right? Seed shape, we will take a seed shape that is round and wrinkled, where round will be the dominant one and wrinkled will be the recessive one. In triad 2, that is the seed color, we can take it yellow and green, where yellow will be the dominant and green will be the recessive one. Okay, so let's cross these. So when we take let's cross these. In dihybrid cross, the parent we can take it as round yellow and wrinkled green. Okay, so these are the parent plants. Round yellow, capital R, capital R, capital Y, and in wrinkled green it is small r, small y, small y. So this is the parent genotype, and after that what we have to do, we have to find out the gametes, right? The parent gametes. Here capital R, capital Y, this will be the gamete, and over here it is small r, small y, that is the gamete. Then we need to fuse this, right? When we are fusing these two, what we will get? Capital R, capital Y, small y. This is the F1 generation that we are getting it. And how we will write, or what is the phenotype that we are writing here? This is round yellow. Why it is round yellow? Because R is dominant and Y is dominant. That is, they are in capital letters, right? So that is, we can write as round yellow. Okay, now next is we need to find out the F2 generation, right? So to find out the F2 generation, what we have to do? We need to self cross this plant. Okay, so we will self cross the round yellow plant, and when we self cross it, what are the possible gametes that we are getting it? Capital R, first we will do the capital ones, right? Capital R, capital Y, then capital R, small y. Then we will take this small r first, small r, then capital Y, small r, small y. So these are the possible gametes for the F2 generation. Okay, this is a Punnett square. Here I am writing capital R, capital Y, then capital R, small y, small r, capital Y, and small r, small y. Okay, in that way, same way we will write here also capital R, capital Y. Okay, then capital R, small y, small r, capital Y, and small r, small y. Next step is we need to cross these. Okay, so first we will cross these two. What is the resulting one? Capital R, capital Y, capital Y. Okay, now next capital R, capital Y, small y. Then capital R, small r, capital Y. Then capital R, small r, capital Y, small y. Right, capital R, small r, capital Y, small y. Next, these we will do it. Capital R, capital Y, small y, small y, small y. So these are the one which we have completed. Okay, now capital R, capital Y. So here we have to write the phenotype. What are the possible phenotype here? As all are in small, capital letters, so this is round yellow. Okay. Here also this is also round yellow. Why it is round yellow? Because capital R, right? R is capital, Y is capital, so it is round yellow. Okay, this one also round yellow. Now here what you can see, R is capital but Y is small letter, right? For R we can write it as round, but Y is small letter, right? So when Y is small or recessive, what we can write? This is green, so round green. Okay, green is associated with the small y. Okay, here round yellow. Okay, now capital R, small r, this is round. Small y, small y is recessive, right? So it is green. Then small r, right? This is recessive. Which one is recessive? Wrinkled one. Okay, so this is wrinkled yellow. Then small r, this is also wrinkled yellow. Then this is round yellow. Small capital R, small r, one is dominant, so round. This is small y, right? Small y, that is recessive. Small y related to green, so round green. Then capital R, sorry, small r is recessive, that is wrinkled. Capital Y, this is yellow, wrinkled yellow. And small r small, these are all recessive, so we can write wrinkled green. Okay, this is what we got it. We have round yellow, then round green, then wrinkled yellow and wrinkled green. So these are the phenotypes that we got it. Okay, now how many round yellow are there? We have 1, 2, 3, 4, 5, 6, 7, 8 and 9. We have 9 round yellow. Then round green, how many round green are there? This one, 1, 2 and 3. We have 3 round green. Then wrinkled yellow, 1, 2 and 3. We have 3 wrinkled yellow. And how many wrinkled green are there? 1. Okay, so what is the ratio? The phenotypic ratio is 9:3:3:1. Okay, so for dihybrid cross, the ratio is 9:3:3:1. Whereas for the monohybrid cross, this is 3:1. Clear? Now you can try this question. A number of times it has come in the board exams also. So in a genetic experiment, pure tall pea plants with white flowers were crossed with pure dwarf pea plants having violet color. Violet flowers. Now mention the different combination of gametes formed by the F1 plants during self-pollination. You have to find out what are the combination of gametes that is formed by the F1 plants. And after that, a total of 256 plants were produced in F2 generation. Show the ratio in which these traits are independently inherited in these 256 plants. So just find out what is the answer and comment in the comment section below. Now, Greco John Mendel has found 3 laws of inheritance. The first law of inheritance is law of dominance. Now, what is law of dominance? When 2 contrasting alleles are present together, only one allele expresses itself, and the expressed allele is the dominant one and the hidden allele is the recessive one. Like for example, in law of dominance, one of the best example is the monohybrid cross. So in monohybrid, we have already seen that a dominant allele is there and a recessive allele is there, right? So what is law of dominance? When 2 alleles are present together, only one allele expresses itself. Okay, and the expressed allele is the dominant one and the hidden allele is the recessive. Okay, now next is law of segregation. Now what is law of segregation? Law of segregation states that 2 alleles of a gene separate. Okay, that 2 alleles of a gene separate from each other during gamete formation, so that each gamete receives only one allele. Like same as an example of monohybrid cross, we have seen that the 2 alleles, right? Capital T, small t. So these will separate and then they receive only one gamete during the gamete formation. Okay, so the law of segregation states that 2 alleles of a gene separate from each other during gamete formation, so that each gamete receives only one allele, and it is also known as the purity of gamete. Now next one is, so law of if you take an example of these two, law of dominance and law of segregation, monohybrid cross is the best example for this. Whereas for the law, what is law of independent assortment? In law of independent assortment, the alleles of different traits are inherited independently of each other. That is an example of dihybrid cross. Okay, in dihybrid cross, the traits are independently inherited, as you have seen earlier, right? The round yellow and wrinkled green. So they are inherited independently for each other. So these are the Mendel's law of inheritance. So there are 3 laws: law of dominance, law of segregation, and law of independent assortment. Okay.